Thank you very much for that kind introduction. Yes, as Gleb said, the title of my talk today is on the advances of ultra-high field MRI for doing neuroscience. And in terms of human imaging, when we talk about ultra-high field, we're really talking about 7 Tesla MRI and above. And I'm here at the Melbourne Brain Centre Imaging Unit, which is a node of the Australian National Imaging Facility. I'd just like to acknowledge them for funding my salary and the instrumentation that we have together with the Victorian biomedical imaging capability in the Australian government. Before I go on, I'd just like to acknowledge the traditional owners and of Australia, and their elders past, present and emerging, and in particular the Wurundjeri people of the Kulin Nation here in Melbourne. And what I'm going to talk about today is really our efforts to push the technology in magnetic resonance imaging to allow for more precise and quantitative bioimaging, in particular of the human brain. And because many of you out there may not be that familiar with how MRI works, I thought I'd just start with a couple of introductory slides about MRI. And basically, MRI instrumentation consists of a rather large static magnet here, and we often refer to this as B0. And this is a picture of a clinical scanner. The state-of-the-art clinical scanners operate at 3 Tesla. But in order to get an MRI image, we need to combine it with some other magnetic fields, namely rotating magnetic fields or magnetic fields rotating at radio frequencies, and some what we call magnetic field gradients. And these are all contained within the setup here. And then we have some receive coils, which are like pickup coils that get the signal back from the human body or the brain in our particular case in neuroscience. And here's a picture of sort of how our magnetic field gradient coils are situated in there. And these gradient coils allow us to encode spatial information in our magnetic resonance signal and allow us to get the images that we get. And what we're really doing when we do magnetic resonance imaging, well, the original experiments are derivatives of nuclear magnetic resonance. And what we're trying to do when we put someone in an MRI scanner and seek to do some neuroscience is that we're trying to make all the different parts of the human brain operate like tiny little mobile phone towers that operate at radio frequencies. And there's some quirks of quantum mechanics that allow this phenomena to occur, but we can do it with great precision. And when we get these parts of the brain to operate like little tiny mobile phone towers, they can tell us a lot about the biology that's going on around them in terms of molecular dynamics and the chemical environment that they're in. And the MRI works on the phenomena that protons or water molecules, or the hydrogen atoms in water molecules, are very, very slightly magnetic or slightly paramagnetic. And that means when you put them into an MRI scanner, they somewhat align with the magnetic field. They don't align perfectly like, say, iron filings would, but they generally are aligned with the magnetic field. So we get this thing that we call the net magnetization. And while we get a lot more signal if water protons were a lot more magnetic, because they're only partially magnetic or weakly magnetic, they have some quirky phenomena that we can make use of to do the nuclear magnetic resonance experiment and even magnetic resonance imaging. And the phenomena that I'm talking about is that these, at the quantum mechanical level, these protons resonate or they rotate about the main magnetic field at very high frequencies in the radio frequency window. And that allows us to detect these radio frequency signals, and they're modulated by their physical and morphological environment and even their chemical environment. And so we can pick up signatures that tell us a lot about the biology that's going on. And in the magnetic resonance experiment, we combine, as I indicated earlier, several different types of magnetic fields to get an image. And we play these magnetic fields out in kind of like music scripts. We play them out in what we call pulse sequences. And we often refer to the MRI scanner as an instrument, and it is basically that. It is a very expensive musical instrument, electrical musical instrument, if you like, but instead of music coming out, we get beautiful images of human biology. And here I've got a little cartoon of our representation of water protons spinning around, or the magnetic dipole spinning around inside the magnetic field. And we can get them to do this in a certain fashion. And we collect the signal in sort of oscillating sinusoids at the radio frequency. And using some 1820s mathematics, namely the Fourier transform and some modern computers, we can convert these oscillating sinusoids to images that represent the human brain. And I've just got a little video here. So when we're talking about 7 Tesla, we're talking about a very, very strong magnetic field. Here's Shauna and my former boss, Roger Ordich, demonstrating just how strong these magnetic fields are. And so they're taping here a bulldog paperclip to the end of a plastic chain, just to show you the strength of the magnetic field. And Shauna's a very experienced radiographer. She knows what she's doing. She can hold it here and whoops, no, not that experienced. So these are extremely high magnetic fields that we're talking about. So high that that you do feel at 7 Tesla when you're going into the magnet, the effects of these static magnetic fields. Now, what happens to the MRI experiment when you push to higher and higher fields? So we push from 3 Tesla, which is the state-of-the-art clinical ones, to 7T. The main basis of going with this higher field is that our MRI signal goes up pretty much linearly with magnetic field. And because the protons are only weakly magnetic and the signal in NMR or MRI is relatively small, we always consider ourselves to be signal to noise deprived in an MRI experiment. And our resolution of our imaging and our ability to quantify the signal in MRI is really dependent on the base signal to noise of your system. And so the easiest way to get more signal to noise is to go to higher and higher fields. But there are some other things that happen that we can also take advantage of too at higher fields. So the chemical shift separation between water protons and other protons of interest, so the hydrogen protons on organic molecules, key neurochemicals in the brain increases. And that allows us to do some really interesting, what we call chemical shift imaging. Our spin and spin lattice relaxation, so how long the signal hangs around for after we excite the protons in our system, changes at high fields. And this can be an impediment to some of our imaging, but we can also exploit this to understand the molecular and morphological environment that the water molecules are in, and that can tell us something about biology or disease that we're interested in. Unfortunately, there's some downsides, like with going to all sorts of high technology, we don't get everything for free and we have to pay some penalty. And our radio frequency deposition into the human body becomes heterogeneous. The interaction of the brain with the magnetic field becomes quite heterogeneous, and we have to deal with this in some way. And the other thing that goes up is the safety requirements gets a little bit more hairy at the higher fields, and we have to take into account the increase in heating and other biological effects at high field. But when we do the experiments correctly, we can really see advantages in improving our traditional MRI sequences that we do to allow us to get much higher spatial resolution. We can go to higher temporal resolutions in the case of functional MRI in terms of being able to image the dynamics of how the brain works during different cognitive exercises or experiments. We can improve and make possible the quantification of neurometabolites, brain iron, and even image directly what we call ex-nuclei, or mostly we're doing direct sodium imaging in our lab. So I indicated earlier that the way we acquire MRI information is to play out these sequences of radio frequency and gradient magnetic pulses. And we call these in MRI science MRI sequences. And these determine the type of information that we can get from our images. And so at 7T, we're blessed by being able to have a huge array of different MRI sequences that allow us to do all sorts of different things with our MRI scanner. So we have the traditional sort of structural MRI scans that produce beautiful anatomical images. And we can do T1 weighted imaging, some imaging where we null the fluid so we can get extremely good gray white matter contrast. We can do T2 imaging that gives us, again, great gray white matter contrast, but a little bit different to flare. We can do diffusion MRI where we can really map the connectivity of white matter throughout the brain. We can measure the dynamics of the brain using a method called functional MRI. We can do this at lower field strength, but we can really speed up the acquisition of these fMRI scans at 7T. And we can start to really do true quantitative metabolic imaging using magnetic resonance spectroscopy, which is directly related to nuclear magnetic resonance spectroscopy, but in this case, directly in the human brain. And we can start to map other things that are biologically important, like the actual magnetic susceptibility of the brain tissue itself, which we can look at the iron quantification in the brain. We can image directly the sodium nucleus and do sodium imaging to understand the importance of sodium in the biology of the human brain. We can look at blood flow, and we can start to look at really, really high resolution angiography, which is important for our stroke neuroscientists. So here's some typical imaging. So we get just really, really, really exquisite high resolution imaging at 7T. And here we really push things to get true 3D 0.6 millimeter resolution with excellent gray white matter contrast here in of the brain. We can segment this using segmentation tools and really look at sort of with great precision the cortical thickness of the brain, which is great for doing aging and traumatic brain injury studies, and even MS and other pathologies. And here again, you just see the beautiful high resolution images that we can get. Now, of course, we can get these images at lower field strengths, but we wouldn't be able to get this type of resolution and contrast and with the kind of signal to noise that we can get at 7 tester. And here's some 2D imaging. You can really see here, we can push the in-plane resolution, and you can see the substructures of the hippocampus now with these beautiful T2 weighted images. And here you can see the perivascular spaces in the brain, which we think are analysing these are extremely important to understanding traumatic brain injury and the aging process of the brain. Again, here just some more super high resolution anatomical imaging. Of course, we can image some things that are closely related to the brain. Namely, we've been working with a vendor to deliver us dedicated eye coils. And here we can really look and quantify all the major structures in 3D of the human eye without doing any sort of dissection. So this is a living, breathing person, and we can measure the structure of the eye with great precision. Here's some of our spinal work, so just getting a bit outside of the brain, but still neuroscience. We've got the ability to do extremely high resolution spinal cord imaging and even look at the tractography or the connectivity within the spine using diffusion MRI. Here's now starting to look at least a snapshot of the dynamics. Here we've been able to weight the blood vessels based on flow of blood through the vessels. And you can see here that we can start to resolve really, really tiny vessels in the brain. And we're interested in these lenticular striate arteries here. And really these arteries you can't see at field strength below 7T. And so Professor Bruce Campbell's a world leader in understanding lacuna infarcts. And we're working with him to look at the role 7T can play in a better understanding these very small, tiny strokes that people get. And of course, like when we started doing these at 1.5T and even 3T, this type of imaging, we'd have to do these in small, tiny segments and stitch them together. And it was really, really difficult to do. But now we can do these all in sort of one slab and without the stitching together and in sort of less than 5 minutes of acquisition. We can start to do some fancy things with our pulse sequences and make them extremely efficient. So here, this is a sequence that was provided to us by Siemens, but we've been working on how we can use it to quantify multiple MRI biomarkers that we think are important and related to various pathology. Namely, we can get out quantitative T1 maps. We can get the T1 weighted anatomical images, obviously. We can start to look at the spin-spin relaxation within the brain and map the spin-spin relaxation. We can map the magnetic susceptibility and what we call susceptibility weighted imaging. We can take intensity projections to really look at the venograms within the brain. And we can also do some mathematics and actually get quantitative susceptibility maps, which are directly related to the concentration of iron within the tissue. And we have a few neuroscientists here at University of Melbourne that very much have a hypothesis that the accumulation of iron in important parts of the brain are related to Alzheimer's disease. Here again are these quantitative susceptibility maps, just to show that we can do it in true 3 dimensions at quite high resolutions. And when you put it all together, you can see these really fine susceptibility or quantitative susceptibility structures within the different layers of the cortical layers. And here's the MIP again, showing the venograms And so we can also look at brain bleeding and things like that with the susceptibility imaging. The Q-SMART technique is the mathematical algorithms that we put together here at the University of Melbourne to really improve this technology and get rid of quite a lot of the artifacts that appear in this sort of imaging traditionally. The other thing that I alluded that we can do is to really do fantastic tractography, and we can do this at 7 Tesla, where we can really map the connectivity between different areas of the brain by the white matter structure. So here we've mapped the corticospinal tracts, and of course we can do whole brain tractography using MRI, and we can really push the spatial resolution. Usually diffusion MRI and tractography is done with a resolution of about 2.5 to 3 millimeter isotropic, and we can certainly get down to half that in all in all 3 directions. You can see this animation here. This was provided by my colleague near the Strict, who did a PhD in high field imaging of MS disease. And here's the beautiful tractography maps that she was able to produce. And you can see that we can zoom this. There's some super resolution techniques as part of the MR-Trix package. You can really zoom in and look at all the crossing fibers and bending fibers within key parts of the brain. Another project we've been working on is to really exploit functional MRI. And this was a project that I collaborated with the Stentrode team. I call him Professor Tom Oxley here at the University of Melbourne. And we shared a PhD student, Peter Yu, where they're developing implantable electrodes for the brain that they hope to be able to get, hopefully, patients who've got who've lost the use of either their arms or legs and have the ability to control exoskeletons or wheelchairs with sort of natural thought. And really this is an ongoing effort on their part and a lot of investment. But in this particular project, we really wanted to see, could we map out the key important areas of the brain that are involved in how the brain processes our intent to execute a motor movement. Because even when you start to think about moving, even if you don't want to move, you start to stimulate the primary motor cortex. So we wanted to image the dynamics of the human brain non-invasively in order to understand where their team should strategically place their stentrodes going forward in their human trials. And the great thing about 7T is that we've been able to really push the spatial and temporal resolution of fMRI. And here we can show that we can look quite specifically at the cortical layers involved, at sort of temporal resolution of 500 milliseconds with a sort of a partial brain coverage over the motor cortex with quite good fidelity. And we get these really quite nice looking images, and we can get these, we can get whole brain images at sort of 1.5 millimeter resolution in sort of 800 milliseconds now, which sort of gives us a tremendous scope to be creative with our cognitive neuroscience that we can do with fMRI 7 Tesla. Here's some of the work that Peter did, and you can see that we've really been able to really specifically map out key areas of the motor cortex. And you can see these very tiny changes in MRI signal that are related to the cognitive task that the volunteer has been put through. Traditionally at 3T or 1.5T, you can't even see these increases in signal. You have to have quite a lot of repetitions and mathematical models to tease out statistical correlations. But here you can see at 7 Tesla that we have, you know, between 5 and 10% change in MRI signal with brain function. We can exploit this increase in fMRI contrast to noise because the improvement in contrast to noise is really an increase in the effect size. And because fMRIs are really a statistical technique, this increase in effect size means that we can really reduce the number of subjects and keep the power of our analyses the same. And so here's a comparison that Professor Ben Harrison's group, or a cognitive neuroscience group in our university, and they've shown that they've been able to produce the same sort of cortical networks for a very specific fMRI experiment, down from 96 subjects to 28 subjects. And so even though 7T MRI is a little bit more expensive to do, recruiting subjects is even more expensive, especially if you've got disease cohorts that are really hard to recruit for. I'm looking forward to the next phase of our research. This is some very preliminary data that's provided by a new PhD student of ours, where we're really trying to do real-time neurofeedback fMRI, and we've embarked on investing in some technology where we can really start to, in real time, feedback the change in fMRI signal to volunteers while they're in the MRI scanner, and get them to see if they can modulate these changes in fMRI signal while they're in the magnet, and see if this can improve the training for different cognitive tasks. I'm just going to change gear a little bit from brain function to now brain metabolomics. And the great thing about MRI is that we can also, within selected regions, we can get fingerprints like spectra that allow us to determine the concentration of key neurometabolites in the brain. So we can, at 7 Tesla, we can start to resolve quantitatively GABA, glutamate, glutamine, glutathione, myoinositol, NAA, and so we can start to test some hypotheses of molecularly driven disease or brain function. One of the ongoing research activities I've been involved in many years is to understand the role of glutamate in seizure-related diseases causing seizures, everything from epilepsy to brain tumors and stroke. And we've had this ongoing hypothesis that the overproduction of glutamate in tissue when it's diseased can cause an increased susceptibility to seizure. And this is a project we started off looking at tumor-related seizures, where we could look at quantifying glutamate. And here was a study of 3T we started out, where we validated the MRI quantification of glutamate versus HPLC from excised tissue of brain tumors. We've now pushed this to an imaging technique now, where we use a thing called Gluces, and this enables us to quantify the dynamic exchange of protons between water and NH groups on the glutamate molecule, and allow us to quantify things that way. And the beauty of this technique is that we can image this in quite high resolution. So this is a very insensitive technique, so the differences in signal we're talking about are fractions of a percent. So we need more and more signal to be able to do this, and so it's obvious application of high field. Here's just a little sort of calibration we did, showing that we get this linear increase in glutamate with assessed signal. And we started to do this in brain tumor patients, and we've embarked on doing this in stroke patients as well now, where we can now map the relative concentration glutamate through the brain. We'd also be able to want to be able to measure the dynamics of sodium, and these next few slides come courtesy of Yasmin Blunk, who's a principal investigator in our group. She spent her PhD developing new MRI technology for imaging directly sodium in the human brain, and has now got a team of PhD students working on further improving this technology. But we've long had a hypothesis in the MR community that MRI sensitive to sodium could help us to better understand pathological processes, either early in disease or even looking at the integrity of tumor cells and early changes in tumor cells as a result of chemoradiation therapy. And here's some, we have to remember that sodium signal is like approximately 10,000 times smaller than the proton MRI signal. So while these may appear to be low res compared to the proton imaging, these are pretty incredible compared to what I've been able to do at lower field previously. And so we really hope to exploit this with different, looking at aging, traumatic brain injury, potentially brain tumors in the future. I'm sort of getting towards the end of my talk, but I thought I'd like to just share with you a little bit of the underlying technology and the challenges we have at 7T. So as we go to higher and higher field to really exploit this signal, we have to go to these tremendously complicated electrical circuit technology to really get the water proton spins, if you like, or to do what we want them to do. They are temperamental and don't like to do what we want them to do all the time. But with so here we use an 8-channel transmit coil here at 7T and a 32-channel receive coil. So we have sort of 8 very complicated circuitry just to drive the transmit radio frequency pulses, and then another 32 of these on the receive side just to get that imaging. And the reason we need to do this is that at 7 Tesla with our traditional RF coils, you can see this really extreme heterogeneity of the distribution of the RF power into the brain. When we go to PTX, we just with hardwiring the electronics in a smart way, we can start to move this distribution around, but it's still highly heterogeneous. So we can move it around to increase our signal in some pockets, but we're hoping to start to deliver on this technology that was proposed out of MGH, I think originally the spokes technology, to really be able to flatten out our radio frequency distribution and allow for sort of getting back some of the sort of shading issues that we have here at 7T. And once we do that, this will really open the doors for a lot more quantitative MRI, because being able to have a flat homogeneous transmit field just makes quantification so much easier. Here's a little bit of our efforts to shim this B1 out, and we can start to see that we can start to move around this heterogeneity in our transmit field in line with while the patients are in the scanner. And here we have like the signal drop, beautiful signal in these 3D C2 weighted images through most parts of the brain, but we can see the dropout here in the cerebellum. When we apply this sort of newer technology, we can start to see this coming back. And this is an active area of research for us. Just a huge shout out to the main members of our team, my director, Professor Lee Johnson, and her team, who are really pushing hard to develop our own pulse sequence technology to further improve 7T MRI. Our 2 radiographers, our chief radiographer, Beck Laren, and our senior radiographer, Braden Tighe from Royal Melbourne Hospital. Muta, who did her PhD with, now principal investigator with us. Yasmin, who's also started out as a PhD student, has been really pushing hard on the sodium imaging. Katie Davies joined our group to look at reconstruction techniques in MR from engineering. Rob Williams, who's really motivated me to get cracking on imaging traumatic brain injury, and has been a co-CI of our US Department of Defense grants and a source of inspiration to really push forward with pragmatic imaging. And Georgia, who's come on board to manage us all and make sure that we're well financed and well looked after in the lab. So we appreciate all her efforts. And with that, I'll finish up with my acknowledgements. I have to acknowledge our Siemens. We're partners with Siemens. I have nothing to declare because I have no financial interest, but they do have a research agreement with us to provide us with state-of-the-art technology before it's released as a commercial product. Our partners around the world that give us access to research and development sequences from Minnesota to Vienna, UPenn, and Moritz in Germany. My clinical and neuroscience collaborators, Terry O'Brien, his group for the work on glutamate, Ben Harrison, his neurocognitive group, have been fantastic supporters of us. Optometry department, Alison McKendrick doing the eye research. Bruce Campbell and Chris Pentelis are long-time collaborators of mine, been tremendously supportive. Chris Rowe and the traumatic brain injury team and Alzheimer's team, and of course Tom Oxley's team who provided the motivation for this Spentro work. And lastly, and most importantly, our funders, the Victorian and Australian governments, Royal Melbourne Health Neuroscience Foundation, and the two imaging organisations I'm part of, our state one here in Victoria, and the National Imaging Facility. And with that, I'll take some questions out there from the audience.